morning. It's our pleasure to welcome Professor David Baker to come and present here. He's a professor of biochemistry at the University of Washington and an adjunct professor of basically everything else, including genome sciences, computer science, chemical engineering, physics, and you remember them all. A man who doesn't need much introduction, but he works on protein design and engineering and has won numerous awards as a member of the National Academy of Sciences, the American Academy of Sciences, et cetera, et cetera. So without further ado, we would love to hear what you have to say about computational design.
There's a microphone here.
I think that mic should be live.
All right, thanks. It's always nice to come back to Berkeley, and yeah, this building I don't think was here when I was across the street in biochemistry. Yeah, no, I'm actually quite sure it wasn't here. Yeah, but it's neat to see. It's neat. Every time I come back here, there seems to be another 3 biology-related buildings. You know, I don't quite understand how they all can really fit. But so today I'm going to talk about design of proteins, basically, and everything I'm going to talk about has the following workflow. So we start with it starts with a computer design calculation. I'll describe briefly how that works, and for a protein with the desired structure, function, or activity. And then since you design the protein, you know what the amino acid sequence is. And of course, these days, once you know the amino acid sequence of a protein, it's really, really easy to make synthetic DNA that encodes the protein. And then we either assemble it from oligos or we just buy genes, and then we make the proteins and assay them. I put this slide in because sometimes I give talks and people ask me if I ever do any experiments, and if we ever do, I don't do many experiments myself, but if we ever do experiments, then so everything I'm going to tell you about is essentially an experimental result. All right, so how do we design proteins? Well, the simplest possible protein design calculation is taking a protein backbone and doing a search over all possible sequence space. I mean, there's a lot of possible sequences. So for the sequence which has the lowest energy when it's in this structure, this is actually turns out to be a pretty simple calculation. Basically, what you're seeing here is it's a search where at every step we're just picking a random position in the protein, say this one, and then substituting in a randomly picked conformation of a randomly chosen amino acid. So, and then we just do this many, many, many times, and the process converges on a small family of low-energy structure sequences. It's actually a little more complicated than this movie because in practice we have to move the backbone too, because if, for example, we're trying to make a brand new structure, there's no guarantee that there exists a sequence for any particular structure. So, but there's likely to be a sequence that will work well in sort of the structural neighborhood of this one. Okay, so this is where we used to get to up to a couple of years ago, and we would order genes and see whether the protein expressed and whether we made the right protein. But if you think about it, there's a conceptual flaw here because what we're doing is we're finding the lowest energy sequence for the given structure. But if we make that protein, there's no guarantee that there isn't some other structure completely different from this one in which that sequence has lower energy. So proteins, the assumption here is that proteins are folding to their lowest energy state. So if we want to design a sequence that folds up to a state that we want, we have to check at the end of this calculation that this sequence actually does fold up to that structure. And so we can do that. We can do that experimentally, or we can. I think they're shutting people out again out there. So okay, so now we take. But fortunately, we've been working on structure prediction for a number of years. So now we take that sequence and we essentially predict the structure. And we do this. We take the sequence and that we've designed. Now we do this large-scale search of conformational space. This is essentially like ab initio structure prediction. In fact, it's everything I'm telling you about today is using the Rosetta program we've developed over the years, and this is just straight Rosetta ab initio structure prediction. Now, it's kind of interesting, actually. The major use we're making of structure prediction right now is to test our design sequences to see if they're actually predicted to fold up the way we want them to. There's a lot of, the conformational space is very big. This takes a lot of computer time, but we have volunteers from all over the world helping us out through a project called Rosetta at Home. And with Rosetta at Home, the results we get look something like this. So where here I'm showing you is 5 different sort of idealized protein structures. They're ideal because they have perfectly straight helices, perfectly straight strands, perfect loops, and naturally occurring proteins always have some idiosyncratic features because of selection for function or evolutionary drift or whatever. But we're kind of interested now in making sort of the platonic ideals of proteins, and so we can make these things that are really free from defects. We can design them on the computer anyway. So anyway, we get the sequence from that first movie I showed, and then we send it out to people all over the world, around the world, and the results we get back look like this. So each red dot is contributed by a different person running that Rosetta structure prediction calculation. On this axis is energy. On this axis is distance from the design target structure. And you can see that some people end up pretty far from the design and high in energy. Other people end up closer and low in energy. The important feature of each of the sequences which are being tested in these 5 plots, which correspond to these 5 designs, is that the energy drops lower and lower and lower as you go towards the design structure, and the lowest energy points are essentially identical to the design. So this is a pretty stringent check, because now we're checking that basically that on the computer one can find there are no lower energy structures. And then when we make those sequences in the lab, we find that they, in fact, do fold up to the desired structures. They're very, very stable proteins, and the structures are essentially identical to what we designed. These are just superpositions of the design, blow-ups of the design core and the experimentally determined structures. So we've been able to go on and do this now for quite a few different proteins. We can also take a given fold like this one here and make different variants. So we can expand the beta strands, make the helices longer, change the register of the pairing, and so make proteins with very different shapes. And this whole process has worked out well enough that a rotation student can come into the lab and design, basically make a new sketch like this and design a sequence that folds up to that structure, and then experimentally characterize the protein, find out, determine its stability and so forth. And the only thing they can't do during the rotation is to actually solve the structures. And so, in fact, 2 of these structures were designed by rotation students. But again, we can do this with quite high accuracy. So the problem of designing globular proteins from scratch is something that we really have gotten quite good at. Okay, so we can go smaller, and we can do this by introducing disulfide bonds. So this just shows 2 folds where we've got disulfide cross-links holding them together. These are under 50 amino acids. And we can go small, So these are kind of globular proteins, but there are other types of proteins in nature. You're familiar with anchor and repeat proteins, other types of proteins, and these proteins, so there's repeat proteins that exist in nature, but there are also a whole huge family of repeat proteins that likely nature didn't find, or the evolutionary process didn't find. And this just shows, I'm going to show you a couple examples of proteins repeat proteins we've designed, and they're repeat proteins because they just have one module which then repeats and repeats and repeats. And this is, these are superpositions of crystal structure and design model. So we can make these types of structures with very high accuracy. This one actually is, it's not shown here, but it's disulfide stapled at each repeat. So these proteins are likely to be extremely rigid. So for wires and other types of materials, I think they could have some interesting properties. And here's another one here's a comparison of a crystal structure to a design model. I should say these designs were made by a postdoc in lab, T.J. Brunette, and the structures were just solved by Damon Eckhart and Gira Bobha in their spare time while they're postdocs doing something else at UCSF. So we're very excited. I mean, one sort of general theme in what we do is if you think about the number of amino acid sequences for a 200 residue protein, there's 20 to the 200th sequences. The number of sequences which have existed during evolution is probably 10 to the 11th. So the number of sequences which haven't been sampled during the evolutionary process is just massively bigger than the set of sequences which have been sampled and their relatives. So there's a whole new world of things to find, things like this that nature never stumbled upon. All right, so going even smaller, we can think about designing cyclic peptides. And here we can actually enumerate all the states and actually explicitly design for one of them to be the lowest energy. And so this just shows examples of cyclic peptides we've designed that when we solve the NMR structures, and this is work by Vikram Mulligan, a postdoc in the lab, they come out very, very close. So we think there's sort of a rich space of molecules, say from the 7 to 20 amino acid size range, which one can make that nature I don't think really explored all that much at all. And of course, we can do things with unnatural. We don't have to use just L amino acids. This shows something with a mixed L and D amino acids. This is using a bicyclic compound. So I think there's a whole new class of molecules, perhaps new therapeutics or new catalysts, which could be perhaps made this way. And so we have this, I'll be saying a few more words about this later, but we have this protein folding game called Fold It, which grew out of Rosetta at home. And as I'll talk about a little bit later, we're very interested now in self-assembly. So we've now had Fold It players designing self-assembling structures. So in Fold It, and the way these puzzles work is you start with an extended polypeptide chain and with no sequence. And what the players are doing is to bend up that chain into the structures that you formed here and get them to interact with identical copies which are presented. We generate symmetry from whatever they're doing. So while you're folding the structure, it's interacting with other copies of itself. And I really find these quite remarkable. It's kind of like protein art. There's some of you will recognize there's things in nature that look sort of like this one, but there's nothing in nature that I know of that looks like anything like this one. And so we're very excited now. Brian is now, a grad student in the lab, is now making these structures, making these sequences, and we'll see how many of them fold up the desired structure. Okay, so far though, everything I've described is like it doesn't really do anything. These are just new types of structures, and of course, what you and most people are interested in, we're interested in, is making proteins that do things. So first I'll talk about binding. And so say you want to bind a protein target, we think about the surface of the protein like a climber might think of a climbing wall, and so we dock disembodied amino acids against this surface and looking for nooks and crannies where they can fit in really well. And then we have to connect those amino acid side chains with a body, and that's shown here for small proteins which bind the influenza virus, which is shown in yellow here. So this shows two different small proteins bound up against the virus. I should say, once we start designing for function, and indeed even for the structures, only a fraction of the designs work. And this was particularly true in this case, where we had to make many designs. You can tell by the numbers. We had to make on the order of 80 designs to find a couple that worked. Now, most of the others weren't made by bacteria or yeast, and so we don't really know why that is. It could be just that, protein expression is kind of hard to debug sometimes. But when we do get things that bind, they bind for the right reason. So these are structures solved by Damien Eckhart in Ian Wilson's lab. Here's the hemagglutinin here, and here's the binding protein binding to the hemagglutinin, and the structures are really right on. So if you the crystal structure is in one color and the design model is in the other. So these design proteins are binding exactly as designed, and that's true in this case also. So we've now made proteins that bind to not only the region down here I just showed you, the stem region, but also the head region in the sialic acid binding site. And so these are sort of interesting as sort of diagnostics, ways of detecting the flu, as well as potential therapeutics. And this just shows a diagnostic application where we can detect flu virus on paper pretty sensitively is a collaboration with Paul Yeager's group at the University of Washington. And this shows there's a collaboration with Deb Fuller's group at the University of Washington, where if mice get the flu, they get sick, they lose weight, but if this protein is given to them, one of these designed proteins, it's just sprayed into their lungs, they don't get sick. So there's potential here for new therapeutics. Other types of, one can also design binding that's conditional. So for example, this illustrates the design of a pH-dependent binding protein. So the application we went for here was antibody purification. There are many protein drugs which are antibodies, and so there's a lot of need to purify them. Currently, the way that's done is using an affinity column like protein A, which you have to go to very low pH to denature the protein. But here we've engineered an interface, which is shown here, where there's a histidine at the interface. So at high pH, this is blue here, you have quite high affinity binding, but at low pH, at pH 5, not that low, pH 5.5, the affinity is much, much lower as the histidine becomes protonated. So that means you can you can bind the antibody if you want to purify at relatively, at high pH, and then elute it under relatively gentle conditions. And this is looking quite interesting for purifying a number of antibodies of interest which fall apart under the conditions that you normally have to purify them. I'll just get through that. Okay, so one thing that we're very excited about now is designing proteins which block interactions between protein-protein interactions inside cells, which are very hard to probe normally. And I thought I would just list these here rather than to give you an idea, rather than go through them in detail, because this may not be of greatest interest to most people in the audience. But in case, I'm always looking for new collaborations. So if you have ideas, let me know afterwards. So there are two proteins that degrade p53. They're MDM4 and MDM2, and we've been able to design proteins which bind to either just this one or just this one, which is proving to be very useful for deconvoluting the roles of these proteins in p53 degradation. And this is with a group at St. Jude's. There's a whole family of proteins which bind the BCL2, which have the BCL2, which are in the BCL2 family involved in apoptosis. And here we've been able to design proteins that are very specific for individual family members, again, which is interesting to probe what the functions of these proteins, but also possibly as therapeutics. And finally, we've been able to make proteins that disrupt the histone methylation complex, which is interesting for a number of reasons. So I put these here because if there are people in the audience who have protein-protein interactions they'd like to disrupt, I think we're getting pretty good at it. Okay, but what I thought I'd spend a little bit more time on is small molecule stuff, because I thought that might be more interesting. So over the years, we've been working on de novo enzyme design, and so the basic idea of this is we build disembodied ideal active sites, sort of idealized catalytic sites, and then originally we were identifying placements for this ideal catalytic site in known protein scaffolds. Increasingly now, we're building the scaffolds up from scratch, and then we optimize the surrounding amino acids for substrate binding. So this is one of those glasses half empty, half full kind of situations. So on the bright side, we've been able to make catalysts for a number of different chemical reactions, which are shown here. And I'll get to the not so bright side in just a bit. But I wanted to show you one example. So these are all kind of model chemical reactions. And this is something actually I think I talked about last time I was here. So I'm not going to go into it much detail today. But we're interested. So what can you do with enzyme design? Well, perhaps potentially you can make completely new types of pathways. So this just shows a pathway that takes formate, and the idea is you could electrochemically reduce the formate reduces it first to formaldehyde. And then we've designed an enzyme, this was done by Justin Siegel, when he was in my lab, that converts 3 formaldehyde molecules to dihydroxyacetone, which then can go into central metabolism. And the idea is that so formate is essentially providing both the energy and the carbon source in this pathway. So there's again, there's good news and bad news here. So the good news is that this enzyme is active. When we put these three enzymes, express them simultaneously in E. coli, and we make an extract, and this is work done in collaboration with Mary Lidstrom's group, Amanda Smith at the University of Washington, we get clear conversion of formate to dihydroxyacetone, and collaborating with Jay's group here as well on this. Turns out there's some other products that get formed also. So anyways, so that's the good news. The bad news is that we haven't been able to get this pathway, this designed enzyme, and actually these two enzymes normally work on two carbon compounds, the acyl rather than the formal compounds. The activities are too low for us. We haven't yet succeeded in getting E. coli to grow on formate, which is really the goal. So I've had fun talking today to people who putting enzymes into microcompartments. Obviously, a problem here is that formaldehyde is toxic and reactive, so having it sequestered away could be a big help. But this is an example of how you can sort of think about completely new pathways with de novo enzyme design. I wanted to give one more example that's also a year or two old, but I think is kind of a fun one. Chris Ivan, who did this work, is now here as a graduate student in Jay's lab. So I told you we started this, we have this interactive game called Fold It, and I just wanted to show you one example of what Fold It players have done with enzyme backbone redesign. Several years ago, we designed an enzyme which catalyzes the reaction between these two Diels-Alder substrates, the Diels-Alder reaction. And I should say Don Hilbert has recently solved the structure of this enzyme bound to transition state analog, or the product actually, and it looks very, very close to this, which we were gratified to see. But okay, I haven't gone to the bad news about designed enzymes, and Jack can tell you what a lot of the bad news is. But I can probably summarize it in a couple ways. First of all, you can see it very clearly on the units on the axis here, which they should be seconds, right? But they're not. So that's the main piece of bad news about design catalysts. But anyway, I wasn't going to. Okay. So the good thing though about this, again, it's half empty, half full. Although it's a very, very lousy catalyst, this thing clearly did catalyze the reaction, but it did a very poor job at it. So so Chris actually posed this as a challenge to Fold It players, and here's what the model of the enzyme looks like with the two substrates. And the one obvious problem is that as you can guess, the Km for the smaller substrate is absolutely horrible. And so the Fold It players, so we just said, can you get more binding interactions? The Fold It players came up with this big insertion here, and then when Chris made it, it was dramatically better than the original design. And Don Hilbert again has basically taken this Fold It player solution and done directed evolution to make it even better. I was quite surprised to see that, or happy to see that when the crystal structure was solved, this insertion came out to be in the right place. Okay, so now let's focus on the bad news. So the bad news really is that the activities of these designed enzymes are very low. And why is this? Well, native enzyme catalysis is really complex and highly optimized. And there's many things that have to happen. You have to bind the substrate, and selectively stabilize the transition state, and release the product. That's something that's sort of beyond where we currently were and probably still are to do really, really accurately. The other thing which I found, which I didn't like, was that when directed evolution was carried out on some of the designs, not all of them, the mechanism would actually switch. And so, when you're doing design, you want to get it right for the right reason, not get it right for the wrong reason. So what we're really focusing on now is to try and break enzyme catalysis down into simpler subproblems. And the ones I'll tell you about are the problem of nucleophile activation, and then the problem of precise control over binding. And you can imagine how if you can get absolutely very, very precise binding of a substrate and be able to activate residues, activate nucleophiles very effectively, that then you could start putting together more active enzymes. Okay, so so the first example I'll give you is of designing a very highly reactive serines. So serine has a high pKa, it's a lousy nucleophile, and so basically we set out to design catalytic triad active sites, basically starting from scratch. And we used fluorophosphate probes developed in Ben Cravath's lab, which he uses to survey the proteome for new proteases, esterases, and proteases. We used these probes to follow the activity of our designs. And then we solved the structures of these to see whether they were active for the for the design reasons. And so this shows some results that we were very happy to see. So this is a design with a catalytic these are actually different design catalytic triads, which you can kind of make out here. This one's serine, histine, histine. This one's serine, histine, aspartate, and there's even an arginine down here. So so first of all, if you take these, they bind fluorophosphate, that's shown in these bands here. And then if you knock out, it's particularly dramatic in these two cases, if you knock out the supporting catalytic, these catalytic triad residues, you lose activation while the protein's still there. That's what you're seeing here. And then the other thing that I was very happy to see here is that when we solved the structures, this was done in the NESG, the structures, and they're superimposed on the designs, came out almost exactly right. And this was a real step forward because what we've done earlier is make histidine esterases, and we hadn't really mastered holding things in place. So the structures didn't often come out the way that they were designed. But we're getting better at designing really precise hydrogen bond networks. Okay, so the other problem that I talked about is substrate binding, and we really should have tackled this one first. It was sort of for sociological reasons. We thought this problem was solved, so we went with enzymes first, but really we should have started here. And so this is a ligand, and this just shows a binding site for that ligand made up of these side chains here. And this shows what that looks like in spacefill with a tight-fitting cavity around the ligand. When the structure was solved, in Barry Stoddard's group, the ligand was found to be almost in exactly the same place as in the design model, and the design interactions were in place. So again, we can, and I should say after some optimization experimentally, this protein binds with high picomolar affinity. So this is a problem where we can. It's easy to tell whether you've got it right for the right reason because you simply solve the crystal structure. And again, it's a little bit better posed because we don't have any moving parts here. All these things are just where they're supposed to be. The nice thing about the design site, if you get the design right, is you can then rationally manipulate it. I'm not going to go through this in detail, but by controlling the hydrogen bonding groups at these positions, we can manipulate the specificity for a series of related steroids. And so now we're using testing these methods out to make binders to a number of different compounds. These are interesting, for example, people here might be interested in them in terms of sensors for metabolites that you might want to produce by metabolic engineering. And we have some interesting binders for compounds like that. So very interested in collaborations basically in everything that I talked about, but this might be an interesting area. We can incorporate unnatural amino acids into these, both the binders and catalysts. This is work of Jeremy Mills, a postdoc in the lab, showing a de novo design metal binding site, crystal structure of and design model superimposed. This is a binding site for metal that uses a bipyridine residues that's been put in using the Schultz lab methodology. Okay, so the last thing I'm going to talk about today is the design of self-assembly, and I thought I'd put this in because there are a number of people here, I think, who are working on exciting related things. And so this is work of Neil King in the lab, and Neil developed a method for designing proteins which self-assemble into a regular polyhedra. I hope you can see the neat thing about this is these proteins, when you express them in E. coli, assemble with very high fidelity to these cubic structures. And these cubic structures are essentially identical. When the crystal structures were solved in Todd Yates's lab, these structures came out essentially identical to the design models. And this just shows another picture of the designs compared to the models down here, just looking down the symmetry axes of this polyhedron. And this is another design of Neil's that's a tetrahedron. Again, looking down the design models and then the crystal structures. So more recently, Neil and Jacob and Will have been extending this to two-component assemblies. So I'll take you through this one a little bit more slowly since it's not published. Here, imagine two tetrahedra that are inverted relative to each other. And now place two different trimeric proteins on the vertices of these polyhedra. Say a blue one on the blue tetrahedron and a green one on the green one. And now slide them together and rotate them, looking for a way in which they fit together in a very shape complementary way. And then once you've got this, Neil, Jacob, and Will redesign, once they have it, they design the interface using the calculation shown in that first movie to make them interact. And then obviously express the two proteins and see if they form the cage. And these are quite beautiful. I hope you can see it, but these are the design models for 5 of them, and these are electron micrographs. And I hope you can again see that these are entirely homogeneous populations. Oh yeah, that's great. So this one has this shape, and there's another one I think you can see here. So when they make these things, they assemble inside E. coli with very high fidelity to make very homogeneous particles. For several of these, Neil and Jacob can express them separately, and they can be assembled afterwards when you mix them. So if you want to encapsulate things, for example, you could have that thing you want to encapsulate present, then mix them together. Crystal structures solved in Todd Yates's lab, again, are very similar to the design models. So these are the design models, and these are the crystal structures. One of the things that's neat about these is they crystallize really, really easily. Some of these have crystallized within hours of setting up trays in Todd's lab because of the high symmetry. And so again, this is that really pretty one, the design model, and then the crystal structure. And so what's neat about these is since we're designing them, we can put anything you want. Oh, that's not good. Okay. Okay. Anywhere. And so there's all kinds of neat things talked about this morning about some very exciting collaborations about, attaching things on the inside and the outside and making bigger ones, whole different types of things. So yeah, we're excited about what can be done with these. So we can make, we've made some of these on here. Other ones we're currently trying to make now, the icosahedral ones, and you could imagine them being used for as new vaccine platforms, putting things on the outside, for target delivery, putting things on the inside. These same methods can be used to form design wires and layers, and I'll just show you an example of a layer. This is a hexagonal lattice designed by Shane Gonen, and when you make this protein in E. coli, it assembles. This is basically just straight out of E. coli to form these kind of sheets. And when if you simply look at the diffraction pattern of these and calculate a map from that, it fits the model very well. So we don't yet have Shane Gonen is now trying. The student who's working on this is now trying to get a high resolution structure. But you can imagine now attaching things, basically making a whole new set of biologically inspired materials by making things like this. And yeah. So I'm sorry, I've gone ridiculously fast, but I do appear to be done. So this leaves plenty of time for questions. And so, reason I went to I was going fast because I wasn't properly acknowledging all the wonderful people who've done all the work. So Nobu and Rie Koga did the work on the de novo scaffold design. Vikram did the work on the cyclic peptide design. Neil, Jacob, and Will did the work on the nano cages, and Shane did the work on the layers I showed you. The protein binding, Sorel and Tim and Aaron did the work on the hemagglutinin I showed, along with the organ, and Eva did the work on the head region binders, and then I think I skipped all of these poor people because I didn't think you would be so interested in them. The small molecule binding work was done by Christy and Sagar, and not on here is the activated serine work that was done by Sridhar Rajappan in my lab in collaboration with Ben Kravak's group and Chu Wang. And the Niels Alder work was done by Chris Ivan, who's a student here now, in collaboration with folded players. All right, so now you have to ask me lots of questions.
Thanks again for that.
Yeah.
Have you tried making polymers?
Well, yeah, so the wires, the real problem we have is that when we design things, we want to know whether we got it right, so we have to be able to solve a crystal structure. So that kind of confines us to relatively rigid things. So, and you know, if you make something that's, well, you sort of saw the problem with that layer I described. It's really hard to crystallize stuff like this. And so, and that's true for one-dimensional wires perhaps even more. So some of the problems a lot with things like this are not that it's computational design calculations, but it's the experimental characterization. But if you have any good ideas for types of polymers to make. One of the things we're interested in doing that now is applying these protein design computational methodologies we've developed to non-natural backbones. I described, say, mixing L and D amino acids or using unnatural amino acids.
You think that you could make, that on the computational design side of it, would be reasonably possible to make a polymer that you could induce polymerization?
Well, that would be kind of a chemistry problem. Like, it depends on what the mechanism of polymerization is. I mean, you have to have some way to induce. I mean, right now, the way that I've described control here is you could have a polymer that's made of two different components that assembles when you mix them together. Yeah.
Sure. You have designed proteins, I guess, de novo around 100 amino acids or so.
Yeah.
How important? Two questions. So one is how important do you think it is to be able to just design a protein that's, say, 400 residues? Would it be easier to make a faster catalyst if the protein's bigger?
Yeah.
And how far away? I mean, I imagine complexity goes up fast.
Yeah. Well, like these repeat proteins are arbitrarily large, so you can just keep making them longer and longer, right? But that's not really a fair answer. We do see when we go up above 150 amino acids that you start getting degeneracies. Like, so if you try and make we're making bigger versions, like proteins with 6 beta strands and then 3 helices on both sides. And then what you can get are the strands sort of reversing order. So there are some more degeneracies as things get bigger. As to whether, I mean, I think the main thing you need to do to have an enzyme is, you know, you need to have a pocket in which, or ligand binding. So we're working on sort of smaller domains, smaller topologies, de novo, which will have pockets. Most of the de novo ones I showed didn't. They were more spherical. And I think that's what we'll need to. Jeremy.
If your guy named Hoffman is still alive, he'd be asking you about what principles, how could you design in the appropriate flexibility? I mean, if you design a rigid design based on a pocket and a side chain interaction and everything, and don't build in the dynamics, you're not going to be able to get such a great enzyme.
Yeah, well, that's a really good question, and I think that's kind of at the frontier of basically protein design right now. And so up till now, basically everything we've done, we want precision, we want everything to be locked down, we want everything to be where it is. But it's totally true that, like an enzyme, you're going to need some flexibility. And I think basically the way to think about it is, instead of just having one really low energy ground state, we're going to have some other low-lying states that the protein can access. And I guess my feeling is we've got to be really good at getting that one ground state consistently before we can start worrying about the others. But I think we're getting close to that stage now, and I think that's absolutely one of the next steps. Yeah.
On the note of static structures and things you can do, and I don't know if you kind of addressed this in the first question, but obviously you have a pocket, right? But sometimes enzymes control the way that a substrate enters with the rest of the protein via the substrate channel. Have you guys tried optimizing sort of the entrance and exit strategy of substrates and products?
Yeah, well, that's another thing which we really haven't really addressed. I mean, we've been, like for these ligand binding things, basically, that would be particularly true for smaller substrates like gases. And so those are things we're looking at sort of in the context of sort of helping people redesign naturally occurring enzymes. But on the de novo side, I think that's still a step away from what we can probably take on realistically. Yeah.
So you mentioned this from the small molecule binding in a protein. Do you see that as inherently the same as enzyme binding substrate, or do you think that these are different problems that should be approached?
Well, no, I think they're analogous problems. They're not the same problem, but again, like if you design an enzyme you got to be able to bind the substrate in a binding mode where it's coming. You got to really hold it up against the catalytic residue. Say like take a protease. You got to jam the protein up against the serine. It's not going to want to go there. So you really need tight control over how the substrate's going to bind. Catalysis can be a matter of a quarter of an angstrom. And so by looking at this ligand binding problem, we can actually learn how to get control over things on that length scale. So I think if you can't control binding, orientation, and geometry to within half an angstrom, you're going to have a hard time making a really good enzyme. So, yeah.
Do you have any trouble expressing these proteins that you're making?
Yeah, we have a lot of trouble expressing these proteins that we make. So, when you design something and you can't express it, you don't really know whether you just made a lousy design or whether the bacteria just didn't like it. So we're very interested. We aren't doing any in vitro protein synthesis, but that would be an interesting route to explore. I mean, it could be that our success rate would be way higher. And we have some evidence that's true because even some proteins that I design, we do find that sometimes they make designs that are like they express at very, very low levels, but when you purify them, they're just rock-stable proteins. So they're just something that they're toxic to bacteria for some reason. So, yeah, so alternative protein synthesis methods can be really powerful for what we're doing. Yeah.
Have you looked at the differences in already in the amino acid composition of designed proteins versus ones that are naturally evolved?
Yeah, well, so we really overdo it when we design proteins, particularly de novo. So the surfaces of our designed proteins are kind of like glutamate and lysine because you want, they're maximally solvated. So yeah, you can always recognize a de novo designed protein for silly reasons like that. In the core, I don't think there's such obvious differences in composition. I mean, we can obviously make the composition whatever you want. I mean, we can avoid any amino acids you don't want to use and put in ones that, even unnatural ones. But the most obvious differences are on the surface. Yeah.
So I remember at the beginning you showed the ability of your design protein, the predictive ability. Was that at home? And it looked, I don't know what the units were, but kind of regardless of what they were, it looked like they were really, 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 really stable.
Yeah.
Is it possible in that regime, like when you're maximizing stability of protein to design for something that changes its conformation in some way on binding?
Yeah, well, again, you could design for something that had two minima rather than just one minima. I should say in those energy calculations, there's an important thing left out, which is the configurational entropy, which is going to oppose binding. And so that's very hard to compute. And so our approach to that is just to try and design things which have the maximal energetic driving force. And that empirically, that turns out to be enough to overcome the configurational entropy. But I have to tell you, I designed a protein which I didn't show you because if I showed it to you, I probably would have gone on for 2 hours instead of half an hour in my talk here. That the estimated free energy folding is something like 65 kcal per mole. So it's a long helical bundle. After I designed it, I went around my lab telling them that protein design was really easy, and I didn't know why it was taking them so long to do everything. And then they said that what I was trying to do was really much easier than what they were doing, which is true. Yeah.
How good is Rosetta at predicting conformations for
disordered proteins?
Yeah, so that's kind of like that's kind of a poorly posed question. What does it mean to predict the conformation of a disordered protein? Maybe an ensemble of. Yeah, the issue there is, how could you know whether you were right? I mean, that's the important thing with computation that I think gets underappreciated is you really have to make sure when you're going to do a computation that there's a very rigorous way of knowing whether you're right or not. And so Rosetta is very good at producing stuff. I mean, it will produce lots of the computational results, but whether they're valid or not is a whole other question. And I don't think we know enough about the structures of disordered proteins to know whether it would get it, the ensemble would be correct or not.
Did you have to change the energy function for the test assembly?
No, it's exactly, all the interactions are the same. I kind of glossed over it, but it's like what you'd expect. It's van der Waals interactions, hydrogen bonds, electrostatic interactions. I mean, the interactions that underlie essentially all non-covalent stuff in biology are all very simple, interactions. So, yeah, it's the same force field and everything I talked about. Yeah.
So, in terms of ligand binding, you have to think about the really polar ligand.
Yeah.
How do you handle those compared to the two?
They're harder. Yeah. Right. So the ligand, so something that's really highly charged, the problem is it's going to be really well solvated when it's floating around in water. And when you bind it, you're going to have to pull away all that water, and you have to pay the cost of desolvating the ions. So we have not attempted to make binders for really, really charged ligands. The ligands that are that are really well suit what we can. The easiest ones are actually ones that have some mixed polar, non-polar character. The problem with completely non-polar things is then you have orientation. But if you have a few, something like the DIG was really good because we had some hydrogen bonds and some non-polar interactions. So very charged things would be tougher. Yeah.
So your de novo structures, they tend to be thermophilic or?
Yeah, they tend to be. I think essentially every de novo design structure we've made doesn't melt at high temperature. Yeah.
Can you model membrane proteins?
Well, so membrane proteins. So I didn't talk about the work we do on structure prediction or structure determination. Yeah, you can certainly, Rosetta has been used, and we do use it to model membrane proteins that exist. The problem with design of membrane proteins is, again, just the testing is much harder, because how do you, and there, you have to somehow assemble it into a membrane. So we really haven't done much with de novo design of membrane proteins, but that's really because just experimentally it's tougher. Yeah.
So we got a couple questions about the difference between design proteins and natural ones. So when you directly evolve them, do they take on characteristics of natural proteins, or are they a little weird?
More beautiful, more perfect, you mean?
Yes. Yeah, do they start to have brain pedicle amino acids on the surface and things like that you would see below the lines between?
Let's see. We haven't done much. That's a good question, and we haven't really done that much evolution. We haven't really done any direct evolution of these completely de novo design things because there hasn't been much to select for. But last time I saw Randy, my thesis advisor, I explained to him that, well, biology is kind of a historical science. It's just stuff that happened to happen, and we can kind of look at it, and just the way you can look at modern day world and try and understand how it got that way and turn over every rock and see what's there. But there's this whole much more beautiful sort of post-evolutionary biology where we can take all the insights we've gotten from studying this historical stuff that's happened and, make a more perfect, around molecules, interactions, whatever. I don't think he bought it, but... all right, well, thanks. Yeah, sorry about rushing.